Hello, my name is Leo, and today I will be discussing the topic of my literature review, NHP Herbals, Their Adversities and Effects on Xenobiotic Metabolism. The use of natural health products has significantly increased on a global scale. NHPs comprise a multi-billion-dollar industry with growing demand for new products and innovations. Historically, health products were always regulated as either a food or drug. NHPs did not quite fit into either category. Many novel products with illegitimate health claims were accessible to consumers and poorly regulated. Canadians and public health officials expressed their concern about the lack of adequate natural health product regulations. In 1998, there was much debate in the Parliament about the need for better regulations. The natural health product regulations were established in 2004 by the Canadian government. This was in response to the growing NHP industry and the concern for safety and efficacy of novel products. The Natural Health Product Directorate was established as the main regulatory authority. They became responsible for the oversight of NHP regulation. Health Canada stated that these regulations would ensure that Canadians have access to natural health products that are safe, effective, and of high quality, while respecting freedom of choice and philosophical and cultural diversity. The Canadian NHP regulatory framework has been recognized as one of the most rigorous and thorough in the world. It enforces and ensures good manufacturing practices in the industry, and additionally, consumers now more than ever are encouraged to report adverse events to Health Canada. This ultimately helps to document and better understand interactions or adversities arising from a specific product. Herbal remedies are a subcategory of natural health products. These are quite prevalent today over the counter. A survey conducted by Health Canada in 2010 found that 73% of Canadians consume one or more natural health products regularly. Around 24% of these use herbal products or remedies. Estimated future use of herbals projects towards 37%. These are some herbal products that are commonly used today. The literature review focuses on St. John's wort and golden seal, as these have considerable risk potential for adverse effects and metabolic interactions. This includes pharmacokinetic and pharmacodynamic interactions with other xenobiotics. The use of NHPs can result in adverse events and interactions. Adverse events generally refer to undesirable side effects like headaches, nausea, mania, etc. Interactions refer to alterations of the pharmacokinetics or pharmacodynamics of another drug or xenobiotic. Pharmacokinetic interactions have an impact on the absorption, distribution, metabolism, and excretion of a drug. Pharmacodynamic interactions consist of inhibitions or potentiating effects at the level of the receptor. It is important to note that metabolic interactions can ultimately lead to other adversities or complications as well. Adverse events can arise from individuals self-prescribing or medicating with herbals, especially without the opinion of a healthcare professional. There is an increase in risk for interactions when taking a prescription medication at the same time. Of people taking NHPs concurrently with prescription medications, only 42% disclose it to their healthcare provider. This is quite alarming because these rates of disclosure have remained the same since 2003. Without the opinion of a healthcare professional, an individual may remain unaware of the contraindications and side effects associated with an herbal product. St. John's wort, or SJW, is an herbal remedy that is commonly used to treat depression and anxiety. Depression is quite common and remains largely undiagnosed and untreated today. Of many people that undergo treatment, 10 to 30% do not improve or simply show a partial response. In several clinical trials, SJW extracts have shown noteworthy efficacy with little side effects. These extracts have proven to be just as effective as many conventional antidepressants, like selective serotonin reuptake inhibitors, SSRIs. However, they display this efficacy only in the treatment of mild to moderate depression. Due to its pharmaceutical-like potency, SJW has been referred to as nature's Prozac. Its bioactives, hyperforin and hypericin, are usually what extracts are standardized to. Most of the antidepressant activity of the herb is actually attributed to hyperforin. SJW works similarly to many selective serotonin reuptake inhibitors. Hyperforin and hypericin are involved in the inhibition of reuptake of norepinephrine, serotonin, and dopamine. Inhibiting the reuptake of these neurotransmitters at critical synapses of the brain alleviates depressive symptoms and improves overall mood. SJW has been reported to have mostly benign side effects, the most common being a headache, and other side effects being dry mouth, nausea, fatigue, sexual dysfunction, and gastrointestinal discomfort. There are also rare events and case reports of psychotic and manic episodes after use. Some pharmacokinetic interactions of SJW include the in vitro induction of P-glycoprotein transporter, also known as PGP, and CYP3A4, both in hepatocytes and intestinal cells, via the pregnane X receptor, PXR. There is also in vivo clinical induction of CYP3A and PGP. In two different clinical studies, it was found that many SJW product variations induce CYP3A. Furthermore, it was also found that PGP lowered the bioavailability of some drugs. Many drugs are substrates for these components, so this is of great clinical concern. In terms of pharmacodynamic interactions, SJW can potentiate the effects of SSRIs. The excessive increase in serotonin at neurological synapses may result in manic episodes or serotonin syndrome, which can be fatal. These interactions are of concern for people taking prescription medications like antidepressants, antiretroviral, and anti-cancer drugs. Golden seal is a perennial herb native to North America. It has been used historically as a remedy for colds or respiratory infections. The most thoroughly studied compounds in golden seal are the bioactives berberine and hydrostein. The antibacterial properties of the herb have been attributed to these alkaloids mostly in vitro. Most data available is on the compound berberine itself, isolated and not within the golden seal plant. There is limited clinical data on the effects of golden seal in humans, let alone the long-term effects. High berberine plasma concentrations can cause jaundice and kernicterus in newborn infants. Kernicterus consists of brain damage from high levels of bilirubin. Berberine has been shown to displace bilirubin from albumin in the blood. Therefore, the use of golden seal is contraindicated for pregnant women as it can result in adverse effects. It has been observed that berberine inhibits PGP transporters in vitro. However, this is inconsistent with the findings of a clinical trial in 2007 that found that inhibition of PGP was minuscule and insignificant in vivo. In subsequent trials, the researchers described the inhibition of CYP3A and CYP2D6 activity by as much as 40%. It was also found that hydrostein could bind these CYP enzymes and form metabolic intermediate complexes that further inhibit activity of the enzymes. The CYP3A family of enzymes are critical components of xenobiotic metabolism. They are catalysts of more than 50% of pharmaceuticals today and make up to 60% of the liver's cytochrome P450 composition. CYP2D6 is another important enzyme as it is responsible for the metabolism of around 30% of all medications. PGP is one of many important and extensively studied integral membrane transporters. PGP plays an essential role in the absorption and disposition of various xenobiotics. SJW and golden seal cause disruptions at these integral components of xenobiotic metabolism. This compromises our ability to metabolize drugs in a desirable and safe manner. People at risk are patients taking antiretroviral, antidepressant, anti-cancer, or diabetes medications. It is important to consider that older adults and the elderly have a greater vulnerability to metabolic interactions and adversities. This is because renal and hepatic detoxification capabilities decrease with age. There is always an increase in risk of adversities when consuming herbals with prescription medications. Not disclosing NHP use to healthcare providers may leave an individual unaware of the adversities and interactions involved. In conclusion, there is a need for more rigorous research on the metabolic effects and adversities associated with herbal products. More importantly, there needs to be more efforts towards educating the public on NHP use and the risk and adversities. Finally, I would like to say thank you to Dr. Monk for this special opportunity. It has helped me grow and learn a lot as a student.